Hi, today we'll be discussing key features or characteristics of a population. At the end of this lesson, students are expected to be able to number 1, differentiate the key features of populations, 2, observe and investigate an actual doodlebug population, and 3, characterize this doodlebug population in terms of its size, density, and dispersion pattern. One attribute of a population is size. Population size refers to the number of individuals that member a population. Population size is affected by 4 demographic factors. Number of births or natality, for example, affects population size positively. Another factor that affects population size positively is immigration. Immigration is the movement of individuals into a population, causing an increase in population size. If natality is the contribution of new members of a population, mortality meanwhile reduces population size through death. Such mortality factors can be either density dependent or density independent. Predators, parasites, and parasitoids are examples of density-dependent factors, while typhoons, earthquakes, and even pesticide use are examples of density-independent factors. Notice a pattern over here? Density-dependent factors are mostly biotic factors, while density-independent factors are abiotic factors. The last demographic factor affecting population size is emigration. This process is quite the opposite of immigration since the movement of individuals is now outwards, causing reduced population size. The effects of demographic processes like mortality is more appropriately illustrated through a survivorship curve. This figure shows that if the population exhibits a type 1 survivorship curve, then most of its members survive until adulthood. Humans have a type 1 survivorship curve because of the high parental care. In contrast, a type 3 survivorship curve reflects how most of the members of a given population die at an early age. Many insect populations, for example bees, show this kind of survivorship curve. Unlike humans, insects show little or no parental. Increase in population size can be predicted through age pyramids. Here you can see the 2014 age pyramid of the Philippine population. The left part of the graph depicts the male population, while the right side reflects the female population. This age pyramid shows that the Philippine population is still growing, as bulk of the members of this population belong to the pre-reproductive and reproductive age groups. When the number of individuals per unit area is concerned, we now deal with another key feature of a population which we term as density. This illustration demonstrates human population density in theory. If each box represents 1 square kilometer and each black dot represents a person, this is what the world's population density would look like in 2005 and in 2050. In reality, however, population density is much more heavy in some places than in others. Example would be in urban areas. This all depends on the availability of resources. Dispersion, or the way by which individuals of a population occur at a particular area, is the third key population attribute. There are 3 main types of population dispersion. Clump type shows that members of the population congregate. A school of fish, for example, is of this type of dispersion. Uniform dispersion, on the other hand, is characterized by population members that are evenly spaced with one another to avoid competition. Many plants in man-made systems, say for example planted rice, have this type of dispersion. Lastly, members of populations can show random distribution if resources for their survival are also randomly distributed. Now that you have a keen idea of population characteristics, it's now time to get to know the model organism that you'll be using to review the concepts discussed earlier. Let's do a doodle search. Doodlebugs are immature forms of lacewings. These are insects from the order Neuroptera. This particular species is Myrmelion angustipennis Banks. It was first described and collected from Las Bañas in 1916. Doodlebugs are also known as antlions. These immature insects are sit-and-wait predators that feed mainly on ants that fall into their sandy pit traps. Their constant search for the most ideal spot to form these pit traps forms doodles along the soil surface, hence the name doodlebug. To collect these insects, one needs to find a generally dry area with sandy soil. The presence of shallow holes or pits in the soil are indicative of the presence of doodlebugs. A common kitchen sifter or strainer and gardening hand trowel are useful tools in collecting doodlebugs. All you need to do is to dig pits and sift for doodlebugs. Once collection is done, these antlions can be transferred in a wooden 20 cm by 20 cm observation boxes filled with no more than 2 inches of sandy soil. The doodlebugs will form defined pits after 12 to 24 hours. In demonstrating population size using doodlebugs, one needs to simply count the number of doodlebugs. If 20 doodlebugs were released in the observation box, then the population size is 20. However, the image above only shows 13 pits. This means that there are only 13 doodlebugs alive after 24 hours. Hence, the population size after 24 hours now becomes 13. The doodlebugs that did not form pits are probably dead due to cannibalism. If population density is your concern, then you simply take the total number of individuals and divide it by the area of the observation box, and multiply the resulting value by 100. The resulting population density for this setup is 5 doodlebugs or antlions per centimeter squared. Now that we have size and density covered, our next task is to identify the type of dispersion that this species exhibits. We can use the nearest neighbor technique developed by Clark and Evans in 1954. For each of the 13 doodlebugs, identify the nearest doodlebug and measure their distance from one another. The full details of the procedure for computing dispersion is indicated in your assigned reading. Shown here are sample values of computed statistics of 5 replicates similar to the 20 by 20 setup established. Dispersion can be inferred through the value of R. Since the R values of all 5 replicates shown here are greater than 1, then we can assume that the type of dispersion that this doodlebug species exhibits is uniform. The type of population dispersion can be confirmed by computing for the C value. Since the values show that C is greater than 2.58, then there is definitely a significant departure from randomness, meaning this doodlebug population can exhibit either clump or uniform distribution. However, the R value is not equal to 1, and therefore the type of dispersion is not clump. The next logical option now is that this population exhibits uniform distribution. Now that we have characterized this doodlebug population using key attributes of a population, it is time to take this lesson further. Be a doodle scholar. Try to test the effect of doodlebug size on dispersion or make observations for interspecific computation for space. The supplementary reading material is provided here. Thank you very much for listening.